Hello, biology superstars. Welcome to the SL content for B1.2, all about proteins. There's only 5 points of information here, but sort of a lot to noodle over in this content. So let's get started. All right. The first thing the IB wants you to be familiar with is the generalized structure of an amino acid. So if you watch the intro video for this section, this is the same image that we used then. And the guidance statement, I'm going to get my pen going. I think I'm going to go purple, or this, well, pink is good. Students should be able to draw a diagram of a generalized amino acid showing the alpha carbon atom with the amine group, carboxyl group, R group, and hydrogen attached. So your guidance here is this is what you need to be able to do. You need to draw a diagram of a generalized amino acid. And we are going to use this as our model. And I might even go, I think I'm going to go piece by piece. So the alpha carbon atom is this one in the middle. I'll do mine, I guess I'll do mine right below. I don't think I'm going to run out of space. So there's your alpha carbon. That's right here. The first thing that they talk about being attached to that is the amine group. I'm going to go green for the amine group. That's over here on the left-hand side. So this alpha carbon is connected to a nitrogen. That nitrogen is also connected to 2 hydrogen atoms. That's the amine or amino group. That's this. The carboxyl group, yeah, sure, let's go this nice aqua color for the carboxyl group. There, the alpha carbon is connected to another carbon. That carbon is double bonded to one oxygen and bonded to another oxygen that is also bonded to a hydrogen. That is carboxyl. The next thing that you need to include in your drawing is, let's go dark blue. The R group is the variable group, the thing that is different between each amino acid. And they have that down to the bottom of the picture. This is also called a side chain. And the last thing up at the top is the lone hydrogen. So the hydrogen is here, and on our diagram it's here. So this is what you should be able to draw. If you're asked to draw a generalized amino acid, you never have to be specific about what the R group is, but note that it's there. All right, next point of information is about how these amino acids come together. So condensation reactions forming dipeptides and longer chains of amino acids. If you've already been through some of the content videos, we've talked about these condensation reactions before. I'm gonna go blue. We've talked about some of these condensation reactions before. In this case, you should be able to write the word equation for this reaction and draw a generalized dipeptide. So the word equation is pretty straightforward. If you have an amino acid and it bonds with another amino acid, you form a peptide bond between those 2 amino acids, and the result is a dipeptide. And that is what is shown in this picture over here. So I'm going to erase so that we have plenty of space to look at this. This is your generalized dipeptide, this part down here. And you can see what happened is the OH group of the carboxyl and one of the hydrogens of this amine group were removed to form a water molecule, as they always are in condensation reactions. And what was left was this carbon bonding with this nitrogen. Here's this carbon and this nitrogen bonded together, and you call that a peptide bond. And this is your generalized dipeptide. All right, let's move on. Next point of information is about dietary requirements for amino acids. So there are 2 types of amino acids that you should be familiar with, essential and non-essential amino acids. I'm going to go purple, I think, for this. Essential amino acids cannot be synthesized and must be obtained from food. Non-essential amino acids can be made from other amino acids. So these ones you don't need to get in your diet because your body can synthesize them from building blocks that you get from other foods. You don't need to know any specific examples. This is worth noting, but you should be familiar with the idea that vegan diets require attention to ensure essential amino acids are consumed. Something that the IB leaves out, but that I think is sort of interesting, is the idea of conditionally essential amino acids. Conditionally essential. And that is when, in times of illness, there are some amino acids that become essential because your body isn't able to synthesize them fast enough to produce all of the amino acids that you need. Generally, amino acids are classified by these properties or characteristics, and especially in the HL content, we'll get into this a little bit more. But generally, just be sort of familiar with the idea that there are dietary requirements about amino acids. All right, and let's move on. So also when you think about the way that amino acids join together, I think I'm going to go green for this one. There are an infinite variety of possible peptide chains. So there are 20 amino acids that are coded for in the genetic code, and peptide chains can have any number of amino acids. So you could have a protein that's made up of like just 3 amino acids, or you could have a protein that's made up of like 400 amino acids. And amino acids can be in any order. So the way that this is generally expressed mathematically is if you think that there are 20 possibilities, and you don't know how long the chain is going to be, your variety for putting them together is 20 to the exponent n. So 20 possible amino acids, 20 amino acids, oopsies, I don't know why it does that sometimes, can go, that's the 20, and can go together in any order, and that's the n. So, and I feel like this comes up maybe like on multiple choice, like how do you represent the infinite varieties of possible peptide chains? Maybe once I've seen it, like you've had to actually do this as a mathematical calculation, but I think in general it's just good to like be a little bit familiar with what this formula is representing. All right. Next point of information, and our last one is about abiotic influence on protein structure. So the effect specifically, I'm going to go pink because it's the last one, effect of pH and temperature on protein structure. And you should be familiar with the term denaturation. So the structure of proteins, we talk about a little bit more in the HL content, but in general, protein structure is stabilized by bonds between R groups. So those are side chains. And we'll talk like for SL, you don't need to know what all the different bonds are. But we'll just say that in general, protein structure is stabilized by bonds between R groups. That BT is between and R groups. And we'll make it a capital R. All right. Those bonds can break, especially at high temperatures or very high or low pHs. So if bonds break, then the protein loses its structure, and that is called denaturation. Protein loses structure. All right, and that is denaturation. And since this whole section of the textbook is about form and function, this idea of form affecting function and of abiotic factors deciding the form and the function is sort of where they decided to leave it off for the SL content. So this is like normal functioning protein. Here you have the addition of heat, and you can see like all of those bonds are broken. Bonds break, and protein loses its form. Protein loses form. All right, and that's where they leave it off for the SL content. If you are in the HL course, I will see you in the next video.